This lesson is part of a fun podcast-style series created by the Rubicon Bridge team for IGCSE learners everywhere. Keep listening and learning.
Welcome to the Deep Dive. Today we're jumping into something really fundamental, truly basic to all life.
Yeah.
How things get into and out of cells.
That's right.
It sounds simple, maybe, but it powers everything. How you breathe, how plants even stand up straight. It's all happening constantly at this tiny level.
Absolutely. And for this deep dive, we're looking at, some core material from a biology textbook chapter. Basically the fundamentals of how substances move across cell boundaries.
Right.
Our job, really, is to sift through it, pull out the key ideas, and make it all make sense for you, make it relevant.
Okay, let's do it. So first up is diffusion. This is something that's, it's going on all the time, right? Even if we don't consciously think about it.
Constantly, yeah. Diffusion, it's basically the net movement of particles. Could be molecules, atoms, ions, whatever. They move from an area where there's a lot of them.
Higher concentration.
Exactly, higher concentration, to an area where there are fewer of them, lower concentration. And the amazing thing, it just happens. It's passive.
Passive, meaning no energy needed from the cell.
Correct. It's all driven by the particles' own random movement. Their kinetic energy. They're just constantly moving, bumping into each other, spreading out.
That idea of constant jiggling?
Yeah.
It works. And the example from the source material, the rotten egg one, or maybe perfume, that's a nicer thought?
Yes, probably nicer.
You spray it in one corner, and pretty soon the scent spreads everywhere. That's it, isn't it? That's diffusion. Those scent particles just randomly moving until they fill the space.
Precisely. And eventually, thankfully, in the rotten egg case, they spread out enough that the concentration drops.
Yeah, thank goodness for that.
And what's driving this, we call it the concentration gradient. You can sort of picture it like a slope, an imaginary slope.
Okay.
Particles naturally move down that slope from high concentration up top down to low concentration at the bottom until things even out.
Right, like rolling downhill. So if it's all just random bouncing around, can anything make it happen faster or slower?
Oh, definitely. There are several factors that affect the rate, how quickly it happens. Our sources point out a few key ones. First, temperature.
More heat, more jiggling.
Exactly. Higher temperature means more kinetic energy, particles move faster, collide more often, so diffusion speeds up.
Makes sense.
Then there's the concentration gradient itself. If that slope is really steep, a big difference between high and low concentrations, diffusion happens faster.
Okay, a bigger push downhill.
Sort of, yeah. Then surface area. If you have a larger area for the particles to move across.
More lanes on the highway.
Good analogy. Yes, more particles can cross at the same time, so the overall rate increases.
Yeah.
Like those agar cube experiments show diffusion happens quicker into a cube with a larger surface area relative to its volume.
Right, I remember those.
And finally, distance. Pretty straightforward. The shorter the distance the particles have to travel, the faster they'll get there. So faster diffusion over shorter distances.
Okay, temperature, gradient, surface area, distance. Got it. So how does this apply in, actual living things? Plants. Us.
Oh, it's absolutely vital. Think about plants needing carbon dioxide for photosynthesis. It diffuses from the air, which has a higher concentration, into their leaves through tiny pores, the stomata.
And oxygen goes out the same way.
Exactly. Oxygen, the waste product, diffuses out. For us, it's how oxygen gets from our lungs into our blood cells and how carbon dioxide gets out of the blood cells to be exhaled.
Just following those concentration gradients.
Just following the gradients. And even inside cells, things like glucose, sodium ions, they move across membranes into the cytoplasm, partly by diffusion, just moving to where there's less of them.
Okay. That covers diffusion. But water.
Yeah.
Water's everywhere in biology. It's fundamental. Does it just diffuse like everything else? How does it move?
Good question. Water is special. It moves by a specific type of diffusion called osmosis.
Osmosis. Heard of it.
Right. So osmosis is specifically the net movement of water molecules. And here's the key difference. It's movement from a region of higher water potential.
Higher water potential? What's that exactly?
Think of it as how free the water molecules are to move. A dilute solution like pure water has lots of free water molecules, so it has a high water potential. A concentrated solution, say sugary water, has fewer free water molecules because they're interacting with the sugar, so it has a lower water potential.
Okay, so water moves from where it's more free or abundant
to where it's less free or abundant, from higher water potential to lower water potential. And critically, this movement happens across a partially permeable membrane.
Partially permeable, meaning it lets some things through but not others.
Exactly. It's like a selective gatekeeper. It lets small molecules like water pass through easily.
But blocks bigger things.
Yeah.
Like sugar.
Precisely. It blocks larger solute molecules like sugar.
Right.
This membrane is crucial for osmosis. The source mentions using dialysis tubing or visking tubing in experiments.
Yeah.
That stuff acts just like a partially permeable membrane. It's great for demonstrating osmosis. You can literally see water moving into or out of the tubing depending on the sugar solution outside.
Right, I think I did that in the lab once. It does make it very clear. So, okay, water moves across this selective membrane. What does that do to actual cells? Animal cells, plant cells?
Well, the effects are quite different, mainly because of the cell wall in plants. Let's take animal cells first, like our red blood cells. They don't have a rigid cell wall.
Right.
So if you put an animal cell in pure water, very high water potential, outside water just floods in by osmosis, the cell swells up and eventually it bursts, called lysis. Now put that same cell in a very concentrated solution, low water potential, outside water moves out of the cell.
And it shrivels up.
It shrinks, yeah, acclination. So animal cells are really sensitive to the water potential around them.
Okay, but plants are different because of the wall. Exactly. Plant cells have that strong cellulose cell wall outside the cell membrane. So put a plant cell in pure water. Water moves in, just like before. The inside swells the cytoplasm, the vacuum.
But it doesn't burst. It doesn't burst. The cell wall pushes back. It's strong enough to resist the pressure. The cell becomes firm, rigid. We call that turgid.
But crisp lettuce?
Perfect example. That crispness is turgo pressure. It's what keeps plants upright and supports their leaves. It's vital.
Wow. Okay. And what if you put the plant cell in salty or sugary water?
Then water moves out of the plant cell, down the water potential gradient. The cell loses water, the cytoplasm and vacuole shrink, and the cell becomes limp or flaccid.
Like wilted lettuce.
Exactly like wilted lettuce. And if it loses too much water, the cell membrane can actually pull away from the inside of the cell wall. That's called plasmolysis.
That sounds bad.
It can be very damaging, yeah. It can kill the cell if it's severe or prolonged. The potato strip experiment really shows this well.
Yeah, you mentioned that. Putting potato pieces in different solutions.
And you see them get really firm and stiff in pure water because they become turgid or floppy and limp in strong sugar solution because they become flaccid. It's osmosis in action.
Very cool. Okay, so diffusion is passive downhill movement. Osmosis is water's special passive movement across a membrane. But what if a cell needs something that's already more concentrated inside than outside? Or there's just very little of it outside. It can't go downhill then. Does it just miss out?
No. Cells have a way. That's where active transport comes in. This is fundamentally different.
How so?
Because active transport moves substances against their concentration gradient, from a region of lower concentration to a region of higher concentration.
So it's moving stuff uphill. That sounds like hard work.
It is. That's the key difference. Unlike diffusion and osmosis, active transport requires energy. The cell has to actively expend energy to make it happen.
Where does the energy come from?
Usually from cellular respiration. The cell basically burns fuel, like glucose, to get the energy needed.
But how does it actually move the stuff against the gradient?
It uses special proteins embedded in the cell membrane, called carrier proteins.
Yes.
Think of them like tiny pumps or revolving doors. These proteins bind to the specific molecule, or ion, on one side, say, the lower concentration side. Then they change shape using that energy.
And release it on the other side, where the concentration is already higher.
Precisely. They pump it across against the natural flow.
Wow. Can you give an example?
Absolutely. The source material gives a classic one. Plant root hair cells absorbing mineral ions, like nitrates, from the soil.
Okay.
Often, the concentration of nitrates in the soil water is actually lower than inside the root cells. But the plant needs those nitrates to grow, to make proteins and DNA.
So it can't rely on diffusion.
Right. Diffusion would actually make nitrates leak out. So the root hair cells use active transport. They use energy and those carrier proteins to actively pump nitrate ions into the cells against the gradient.
Forcing them in, essentially. That's pretty clever.
It's incredibly important, and it happens in our bodies too. Think about nerve function there. Active transport pumps are constantly working to maintain the right balance of sodium and potassium ions across nerve cell membranes, which is essential for sending nerve impulses.
So it's not just plants grabbing nitrates, it's fundamental everywhere.
Everywhere. It's a real energy investment for the cell, but often it's absolutely necessary for survival.
Okay, let's recap then. We've got diffusion passive, down the gradient, random movement, osmosis water's special passive movement across a partially permeable membrane driven by water potential. And active transport requires energy, moves things against the gradient using carrier proteins.
That's a great summary. 3 distinct vital mechanisms.
And they're not just, terms in a book. They're literally how every cell in you, in plants, in everything alive, manages its internal environment, gets what it needs, gets rid of waste. It's happening constantly.
Absolutely constantly. And thinking about that, how finely tuned these processes have to be, the right membranes, the right proteins, the energy supply for active transport, it really makes you think.
Yeah.
Well, it brings up a question, doesn't it? If these transport mechanisms are so fundamental, what happens if one of them goes wrong, even slightly?
Like if a carrier protein pump breaks down or the membrane isn't selective enough.
Exactly. Think about the consequences for the cell, or even the whole organism. We see diseases like cystic fibrosis you mentioned earlier, where a faulty transport protein causes massive problems. It highlights just how critical controlling this traffic in and out of cells really is.
That's definitely something to mull over, how dependent we are on these invisible, intricate processes working perfectly. Thanks for diving deep with us today.
Thanks for listening. More chapters coming soon from Rubicon Bridge.